Good evening. I'm Jesse Feldmanstein, one of the supervisors of the Huntington Youth Council. The next presenter is Danielle Fersler. Danielle Fersler is a senior at Northport High School and conducted research through the Breast Cancer Research Laboratory at the Fox Chase Center outside Philadelphia. Danielle sought to determine how estrogens affect breast cancer cells. Her project is titled Effect of 17-beta estradiol on the proliferation of human breast epithelial cells. Without further ado, Danielle Fersler.
Hello, my name is Danielle Fersler, and I'm a senior at Northport High School. And I study the effect of 17-beta estradiol on BSMCF cell proliferation. 17-beta estradiol is an endogenous estrogen, meaning it's an estrogen found within the human body, and it is actually the most potent estrogen from menarche to menopause. And the way estrogens work in the body is they are attached to an ER, or an estrogen receptor, which attaches to a ligand. And what that does is it activates gene transcription, which means it copies the genes. And BSMCF cells, that BSMCF stands for Boyden Chamber Selected Michigan Cancer Foundation. Those cells are derived from a cell line called MCF-10F, which is Michigan Cancer Foundation 10F, which were cells donated by a woman to the Michigan Cancer Foundation. And what they did with them there was they inserted concentrations of 17-beta estradiol to those MCF-10F cells, and they did this for 2 times a week for 3 weeks, and they saw that it changed the morphology of the cells. What it did specifically was it changed the ductiles such that they grew more. And that changed cell is called the TR-MCF, the transformed MCF, which basically, because it's the cells change, they are cancer-like cells. And then what they did was they used a Boyden chamber, which is, I didn't have a picture on there, sorry. But it's basically like there's a cup and there's a porous membrane, and underneath the porous membrane there's a chemoattractant, such that the TRMCF, the invasive cancer-like cells, will be attracted through. And that is what the MCF cells are. Basically, they're the invasive cancer-like cells. So for my experiment, I used a well plate layout with 8 replicates, and what I used was, first there was a column of blank cells, which means they only receive the high calcium media, which is what the cells need for food and such. And then the control received the media and the Almar blue, and the rest of the 3 columns received the 3 concentrations, which I used 0.7 nanomolars, 7 nanomolars, and 70 nanomolars of 17-beta estradiol, along with Almar blue and high calcium media. Almar blue was the assay I used. It's basically an indicator to how much cell proliferation there is, which is basically the amount of cells there are. And Almar blue is resazurin, which is blue in color, and living cells naturally reduce this to resarufin, which is red or pink. And the way I measured how much proliferation there was using this device called an epoch reader. And basically what it did was measure the % reduction of the resazurin to the resarufin, which directly correlates to the cell proliferation, the number of cells, because if there are more cells, then there's more resazurin reduced. So what the epoch reader does is it has basically laser lights that go under each well, and what it does is it shoots the laser light up and it measures the absorbencies at 570 nanometers and 600 nanometers, which is approximately the wavelengths of red and blue. And it uses that to calculate the % reduction. It has a whole normalization formula which consists of the blanks, which is the ones without the Almar blue, such that you know what the absorbencies are normally without any concentrations, without any Almar blue, and the ones with just the Alamo blue, and no concentrations. So the way I actually conducted the experiment was I used 500 cells per well with the concentrations 0.7 nanometers, 7 nanometers, and 70 nanometers. So in the morning, what I would do is I would put the concentrations in and use high calcium meter overnight. And then in the morning, put Alamo blue at a 10% ratio and incubate it for 4 hours. And then use the EPOC reader to measure the cell proliferation. And that was repeated 3 times. We did the experiment. I had 4 weeks at the Breast Cancer Research Lab, and we had the experiment each week. And unfortunately, my results did not show a statistically significant effect of 17-beta estradiol on the cell proliferation. There are several reasons for that. I really learned how frustrating research could be, because things such as there was a contamination in the hood, so we had to throw out that data, and then there was an incubator malfunction, so had to throw that out. And then also, the main reason I really feel the results are not statistically significant was that the time period for the treatments was not long enough. So I'd really like to thank the Huntington Breast Cancer Action Coalition for giving me this opportunity to do the research and to speak to you all about this topic. Thank you.
Are there any questions?
I'm sorry.
For those who haven't done bench-level research, sometimes negative results can be just as important and significant as positive results. But what would be your next step to take the experiment further to determine whether or not there is an effect or there is not an effect between 7-beta estradiol and the proliferation of the epithelial cells?
Like I said, I would definitely do longer treatment periods. Like, instead of just doing the 4 days as I had done, I would try to do maybe 5, 6 days. And one of the things I really didn't like was we had to vacuum it out every day. We had to vacuum out the old media with the concentrations and put in new. And the reason this was very kind of risky, especially for a beginner like me working under this, was because you could accidentally vacuum out the cells as well, which would decrease the amount of cells and would mess up the experiment. So I definitely want to try to find a new or better way to do that part.
Have you thought about using other cell lines, like cell lines derived from BRCA gene, breast cancer gene positive patients that might be more sensitive to the effects of the 17-beta estradiol?
Well, I just did this specific cell line. I haven't really thought about using other cell lines yet.
Because some breast cancers, when they're tested for chemotherapy, are checked to see if they're hormonal positive or negative. In other words, will hormonal therapy work? So some cell lines may not be as sensitive to the effects of the estrogen, and some might be. So a next step might be varying your dosages of the drug, but also trying different cell lines to see if some cell lines are affected by it and some aren't.
Okay, thank you very much for the advice.
Any other questions? Thank you very much. Round of applause.
All right, sorry if I mispronounced your name, but the next person we have up is Josh Solomowitz. Sorry if I said that wrong. He is a senior at Huntington High School and conducted research at the Fox Chase Cancer Research out of Philadelphia. Josh set out to compare the different effects of hormones or proteins to the rat mammary glands that would be able to further differentiate them, reducing the number of terminal end buds and resulting in a decreased chance of being afflicted with breast cancer. His project is titled Effects of Hormones in Relation to the Differentiation of Terminal End Buds.
Thank you. Like you said, my name is Joshua Swamwitz. Before I get into all the boring sciencey stuff, I want to give you guys a little bit about my background. So 2 summers ago, in 2011, I was selected by the HBCAC to travel to Philadelphia for 1 month and study at the Fox Chase Cancer Research Center in Philadelphia. From about 9 to 5, I basically looked at pictures such as this by my right-hand corner, and also had meetings. So let's get into the presentation. The effect of hormones in relation to the differentiation of terminal end buds in the rat mammary gland. This sounds like a mouthful, so I'm going to explain it step by step. So in humans and rats, they have breasts, and inside the breast is mammary tissue. Mammary glands are inside the mammary tissue, and these glands are made up of different structures. There's lobules type 1, 2, and 3, terminal end buds, and terminal ducts. Each portion of the mammary gland has a specific function. The terminal duct actually doesn't have a function. The terminal end bud contains stem cells, and those can differentiate into different types of structures, such as lobules type 1, 2, and 3. The different lobules just take up space. There are lobule type 4s, which we didn't look at. Those hold milk for when the rat or the human is has to feed an infant. But we specifically looked at terminal end buds because they have stem cells, they can differentiate into any type of structure, but it only takes one carcinogen for the terminal end bud to turn cancerous, and then it spreads to the rest of the terminal end buds, and that's basically how you get breast cancer. So we looked at preventing that. So, like I said, we looked at pictures such as these. That's basically a mammary gland with different types of structures. We counted, they were treated with different hormones, such as HCG, progesterone, and estrogen, and then we had a saline control group, which was not tested at all. And we saw which one of these decreased the amount of terminal end buds the most, because terminal end buds are the most at risk for turning cancerous. So after basically looking at a lot of those sheets over the course of the summer, we conclude that HCG was in fact the one that decreased them the most. This was because this is the best time, excuse me, sorry. I was going to say this for later, but we concluded also that there's a window of susceptibility for a rat and human. Rat, it's 35 to 55 days of age, and human, it's from 16 years to 24 years of age. And this is the most common point in their life where one can be inflicted with breast cancer. So we concluded that if one had high amounts of HCG in their body at this time, which I'm not saying go do this, but if a woman did become pregnant during that time, they'd have high amounts of HCG. They were at 1/3 chance less of getting breast cancer later in life. So for future research, we could see possible injections of HCG, not as a cure, but a prevention method. That could, however, damage the body because injecting a hormone into the body can also have negative effects as well. That's the downside to injections. So for future research, hopefully we can find a hormone or a piece of HCG, like a protein group or something, that would have a positive effect on differentiating these terminal end buds into harmless structures and not hurting the body at all. Thank you.
Does anyone have any questions? Thank you very much. Let's have a round of applause.
The next speaker is Colin Francis. Colin Francis is a junior at Huntington High School and conducted research through the NASA Space Radiation Laboratory at Brookhaven National Laboratory. Colin sought to study the effects of radiation exposure on keratinocyte cells found in the surface of the skin. His project is titled An Analysis of P53 Role in DNA Damage Senescence Response to Ionizing Radiation. Here is Colin.
Hello. There's my poster, as you can see. And what we did, we researched P53, which is a gene that helps when a cell is becoming carcinogenous, when it's about to become cancer, P53 steps in and realizes there's something wrong with the cell. The cell is dividing too quickly. Let's shut it down. And the way they shut it down is through senescence, or growing old. The cell, instead of dividing, it just kind of grows fat and dies. And we studied what would happen if you were to remove the P53. And so what we did, we got a sample of cells called Hakat, which it just means they don't have as much P53, or they have defunct P53. This meant that we could hit them with radiation, and instead of the P53 stepping in to stop them from becoming a tumor, they would continue dividing and